coffee, so I think it's probably best to move on to our final speaker this morning is Jan Slingenbergh. Again, an incredible track record of working around the world and with organisations like the FAO. I mean, this morning the speakers certainly have been a tour de force of people who have worked across the world with all the major organisations. So, Jan, you have the final slot between us and coffee, so I'll be up with 5 minutes to go.
Thank you. Let me take you to some livestock pathogens. I would like to take a broad brush perspective and look at some livestock viruses and bacteria. Before that, let me start off and say something we all know. A parasite-host interaction tends to bring damage to the host. Very simple. But that's only one side of it. Given enough time, the parasite and host tend to become closer, with the parasite or the pathogen becoming more host specific. Now, here is an infection plane trying to make allowance for both parasite-host intimacy and hostility, taking infection duration and infection severities as proxies. So we have an infection plane that we wish to populate with pathogens, with viruses. We can select from actually 3 different but related animal type host domains. We have people, we have livestock, and we've got wildlife. Now, we spent quite a bit of time already on wildlife and ecosystems. Let's have a look at the farming landscape. I believe there are good reasons to believe that livestock pathogens, they're very exciting and they're highly dynamic. There are good reasons for this. Livestock pathogens are in fact rather new. It's only been 12,000 years that humans started to domesticate plant and animals and kick-starting the Neolithic agricultural revolution, which brought a gradual build-up of true livestock pathogens, and of course passage of livestock to humans and back again. There are 2 more defining moments, I believe, in the evolution of livestock viruses. One, I believe, took place during the 19th century, the second half of the 19th century, when the demand for animal protein became so significant that it encouraged people to, instead of having scavenging pigs and poultry around the backyard, to confine them and feed them with grain that had become relatively cheap to produce. Now that sort of kick-started a gradual increase of commercial animal protein production. Another big step is 1 century later, towards the end of the 20th century, when following the Green Revolution, the livestock revolution tried to supply the growing world medium-level income class with animal protein products. And this now brings me to the 1990s being of particular interest, because that's where we are at the peak of the livestock revolution, and so we wish pathogens from that period of time. I screened the FAO, OIE, WHO Animal Health yearbook, went through 130 or something viruses and bacteria, and then retained 35 viruses and 24 bacteria, of which we can say these are real livestock pathogens because they have 1 or more of the common livestock species as primary host. So let me recall the hypothesis. I suggested that given time, pathogen and host tend to become more intimate. And we expect this to be reflected in the infection becoming more persistent and also more benign. Now let's distribute these viruses across the infection plane. I will, in a minute, go into individual virus infections and diseases. Let me for now share two important findings. One is that if we aggregate these viruses at the family level, we find over 60% of these viruses actually belonging to 5 virus families. We have to the right the retroviruses, to the far left we got the myxoviruses, and in between we got the 2 DNA families, the pox and the herpes viruses. And these virus families appear to align in accordance with infection duration. That, I think, is of considerable interest. A second finding is that if we look at the names of the infection or disease, we actually also get an indication of organ system tropism and the location of the infection within the body. And this now tells us that epithelial infections are at the acute infection site, whilst at the right side we have internal organ involvement. And in between we've got mixed forms. Now, we have some basis to piece together the trait profile of a specialist virus. We started off by saying that the virus would be more host-specific. Then we brought in infection durability as a fitness trait. We brought in benignity, and now we have the evidence, we have the suggestion that these viruses may actually withdraw in the body environment. And we can build from there and see if also the transmission ecology and the host environment specifics are in fact reflected in the infection strategy, following the infection shedding transmission chain of causation. And in doing so, if a virus hides inside the body, then of course the likelihood of vertical transmission increases. And with it, the vulnerability of this pathogen, should it be exposed to the conditions external to the host body, it will be sensitive and vulnerable. When we speculate on the disease behavior at the herd level, we expect endemic or exotic forms of disease. And then when it comes to the host environment specifics, for a persistent infection, we need a host that, of course, lives long enough. And if vertical transmission becomes important, then of course it does help if the host lives long enough and engages in reproductive behavior to permit the passage of virus from parents to the offspring. So we use this to pull together a specialist score on the basis of the transmission ecology and host environment. And then the score was semi-quantitative, low, medium, high, 1, 2, or 3, with 3 meaning probably more of a specialist. And then putting these scores back in the infection plane, we see a lot of number 3s aggregating in the right bottom corner. Now, let us revisit some of the infections. We have at the bottom right enzootic bovine leukosis. We have swamp fever, which is another word for equine infectious anemia. We have to the left column equine coital exanthema. We've got equine viral enteritis and IBR-IPV, a bovine disease. So we got bovines and equines in the right bottom corner. Let's move up to the left upper corner. We've got avian infectious bronchitis, avian laryngotracheitis, duck enteritis, highly pathogenic avian influenza, Newcastle disease, and then 2 ruminant diseases. And the column to the right, we've got classical swine fever, Gumboro infection, it's an avian disease. We've got PRRS, which stands for porcine reproductive and respiratory syndrome. We've got porcine enteritis, the Teschen virus, and then SECD stands for swine enteric coronavirus disease. And so in between the pigs and the poultry and the bovines and the equines, we've got most of the small ruminant diseases. So basically, if we have a proper look at the specialist score, we note that the infection duration weighs in much more strongly than infection severity in defining the virus as a specialist. If we look at the individual variables, we note that in 11 out of 12 incidents, where we move from the left to the right, from acute to more persistent infections, we have this increase in specialist score. And then if we hold the infection duration against the specialist score, pooling all these variables, as we move from acute to persistent infections, we got a doubling of the specialist score. Now, I'm not trying to impress you with all the numbers here. I think it's more about deductive reasoning. We can say with that there is some evidence to suggest that a specialist virus causes a persistent, not necessarily benign, in the body infection. And I will now move on to some bacterial infections where I did the same thing, and here is what we find. In the middle of the infection plane, we've got a whole number of epithelial bacterial infections. These are the classical contagious livestock diseases. And to the bottom right, we do not see many bacterial infections, at least not known to us so far. And perhaps there are even less bacterial infections here when we compare to the persistent and quasi-inapparent bacterial infections that we do find in wildlife, in particular in rodents. And when we move to the upper left corner, we do find bacterial infections, we do find acute sepsis, but then these bacteria, they carry so much environment-related fitness that they hardly corroborate with the Koch's postulates. So basically, for the bacterial infections, we do not find the specialist, generalist continuum that was suggested by the livestock viruses. Let's pull the viruses and bacteria together. If we take a pathogen ecological continuum that runs from environment-related pathogen fitness all the way to body infection-related pathogen fitness, we have to the far right, of course, the persistent specialist viruses that do display infection plasticity, but in the form of adjustment of infection duration. The more generalist viruses, they are capable of adjusting infection severity. And moving on from there, we have the bacteria, which naturally are found at the mucosal surface in the body cavities and tracts. And so bacteria, in fact, are capable of adjusting their adherence pattern, which is quite different from what viruses actually do. And further to the left still, we find bacteria that have acquired fitness to survive in the conditions that exist outside the body of the host. So viruses and bacteria actually are very complementary in ecological terms. Now, let us ask the question, how does a specialist bacterium actually look like if we wish the same sort of trait profile which we designed for the viruses? It can actually two different opposite direction. An obligate intracellular bacterium, in fact, may go the way the viruses, may go what a persistent virus may do. Whilst oppositely, a bacterium at the mucosal surface might actually integrate with the microbiota, which we find particularly in the enteric tract environment. And so this applies to the specialist bacteria. We can also say that the generalist bacteria will go the opposite way, in the sense that epithelial tract infections may become generalized, say in the enteric tract, or become ascending infections, as we see in the respiratory or in the reproductive tract. And these are, in fact, the classical bacterial livestock infections that are disappearing from most modern livestock husbandry systems, whilst another group is rapidly on the increase, and these are the novel forms of bacteria and also the bacterial extracellular substances that are produced by the bacteria that are found on the surface of the mucosa in the enteric tract environment. And they may not cause much of a clinical infection in the primary livestock host, but then leaking out and contaminating water and food are responsible for a growing number of veterinary public health and in particular food safety hazards. And so before I conclude, let me recall one complication here that I haven't mentioned. The livestock, the host environment, of course, comprises more compartments than just the host body. We do have pattern formation within the host body, population, community, host domain, and even at the level of the geographic range of the disease complex, we see pattern formation. On a positive note, all these different dynamics in all these compartments collectively support the single goal that is written in the genome of the pathogen. And so the pathogen life cycle and the transmission ecology and the host environment, they are, of course, in reciprocal interaction, but they all tell the same story, and things can be inferred from one another. And this now we may apply in risk analysis and management when we are dealing with livestock pathogens. And so, in conclusion, I believe we have good news and bad news. The bad news is that there is a lot more to come still. The good news is that we might actually do something, prevent this, and go back to the drivers. I think we should start asking ourselves, do we really want a broiler chicken to weigh 2.5 kilograms after 5 or 6 weeks? That's, I think, is a very pertinent question. A second question, do we want 40 piglets being weaned by one female pig in a year? And addressing livestock disease emergence at source, at the driver level, brings these questions. At the level of risk analysis and management, I think we may want to ask ourselves, do we really rely just on bioexclusion, or are there options to actually start living with microbes? As it was already suggested, let's go for the friendly microbes. Let's go for the green microbes. And this would open the door for sustainable livestock production. Thank you.
Thank you. That gives us time for some questions. I'm sure the philosophical questions about whether it's right to feed people in this way we can deal with, hopefully, in the half hour that we have at the end. So if we could have maybe one or two more focused questions on the earlier part of the talk.